In prokaryotes, we have the process of transcription. This is going to occur very similarly, at least from a high level, to how it's going to occur in eukaryotes. There are differences, of course, but we're going to focus on the simpler process first. We're going to talk about the different steps involved in transcription. We're going to talk a little bit about control of transcription. These things called promoters are really important in specifying where a gene is to the complex that's going to do transcription, but also when a gene is going to be expressed. So we'll talk about how and when this happens at a high level. Gene expression and control of that is a topic for later sections and also further courses later in your biology career. Okay, so here's our model of a prokaryote, right? We said we have a circular chromosome that is not enclosed in a nucleus. That means that transcription can occur, and at the same time, translation can occur. We'll come back to translation in a later section, but we're going to focus on transcription right now. So transcription is the process of taking information from DNA and making an RNA copy of this. The enzyme that does this is called RNA polymerase. It transcribes DNA into RNA. So remember we talked about DNA polymerase makes polymers of DNA. Well, RNA polymerase makes polymers of RNA. But it's not going to just do that randomly. It is going to use the DNA as a template to build the RNA strand. So you'll notice here I have two strands of DNA. One of them we call the template strand. That is the one that we're going to build the RNA off of. And in this case, I'm going to talk about mRNA. Then we have the coding strand, which is not going to do anything. It's going to sit there. But if you notice, it will read similar to the mRNA that is produced. So remember, RNA is very similar to DNA. It follows our same rules of DNA, the three rules that we talked about. It has complementary base pairings. The one exception is U is substituted for T, so A pairs with U here. We also have RNA as a single-stranded molecule. In most cases, it's single-stranded, and the sugar in there is ribose instead of deoxyribose. But otherwise, it gets built 5' to 3'. It has to be anti-parallel, all of those things. So let's look at making this mRNA here. Okay, this mRNA is made from working off the DNA template strand. So what's going to happen is the DNA is going to unzip, and we're going to build this mRNA using the sequence from this DNA template here. Meanwhile, the coding strand will not do anything during this process, but you will note that it will read the same as what the mRNA is going to read, except T and U are swapped. Okay, so let's look at this. RNA polymerase comes in, and RNA polymerase is a really talented molecule. It does not need primers. It unzips the DNA double helix on its own. It can do all these things. It does all this stuff. RNA polymerase comes in and starts building a RNA copy, basically, using complementary base pairings. So it works off of the template strand. So you can see here, A paired with T, U paired with A, G paired with C, so on and so forth. The RNA is built in a 5' to 3' manner. It's built anti-parallel to the DNA, and this one strand of DNA is serving as the template, and the RNA polymerase is chugging along, building that new RNA. There's no primers needed here, so RNA polymerase can just start adding bases as it needs to. In bacteria, we have some interesting things happening in the genome. We've said that bacterial genomes are much, much smaller than eukaryotes. We look at E. coli, 4 million bases versus 3 billion in humans. That doesn't actually mean that they have 1,000 times less genes, though. A lot of the human genome is not actually coding for proteins. When we look at bacterial genomes, they're very tightly packed and well organized. And we have this phenomenon called operons. So an operon is where several genes that are involved in a similar process are situated right next to each other in the bacterial genome. Here you can see 4 what we call structural genes that will code for proteins that do some sort of catalytic reactions. And then we can see some regulatory genes at the end here. This whole thing is an operon. We have 1, 2, 3, 4, 5, 6 genes in there. Now, each one of these pieces, like MDTA, is one separate gene. But this whole thing is controlled by one region that we call a promoter. A promoter is a sequence of DNA that is going to help basically signify that there's a gene there. Now, some people will say promoters control gene expression. I don't like that term because promoters are DNA regions. DNA does not actually do anything. DNA does not control anything. What the DNA does is DNA gets bound by proteins. So this protein here comes in and binds to the DNA, and it actually controls whether these genes get transcribed or not. This operon, when it does get transcribed, RNA polymerase comes in and it makes one big mRNA. We call this a polycistronic mRNA. It means it has many genes in it. So this whole thing gets made into one mRNA. And then later we will see that each of these little segments, these genes, gets transcribed into a separate protein by the ribosome. This operon structure is pretty unique to bacteria. We do not find this in eukaryotes. So this is one of the ways that prokaryotes have compacted the process. They speed it up. So here we would make a big mRNA here, and one of these regions, MDTA, would get converted into a protein there, but we would have one big long messenger RNA there. Okay, so this question of control. How do we know when to make the mRNA and when not to? That is a very complicated question, and there are many different answers depending on the gene or the operon in the case of bacteria. We will examine bacterial gene control in more detail later. We're going to come to that. We have to understand the basic raw, like what's going to happen though. So one of the key players that controls where and when a gene is transcribed is this thing called the sigma factor. So RNA polymerase is not just one enzyme. We call RNA polymerase a holoenzyme. It has a lot of different pieces to it. One of those pieces is something called a sigma factor. A sigma factor helps RNA polymerase find genes, and it targets specific promoter sequences. So the promoter is a region of DNA in front of the genes, okay? And in bacteria, this is really tightly regulated. There's usually a -10 sequence and a -35 sequence. These sequences are what we call conserved. They have a similar base pair structure. Now, sigma factor is what we call a transcription factor. It's a protein that binds to DNA and regulates transcription. Okay? It's right there in the name. So the sigma factor basically helps scan along until it finds these promoter sequences, and then it binds to that DNA. And it is upstream of what we call the gene, right? So upstream means its base is ahead of where the gene sequence is. So once the sigma factor finds these sequences, it helps bind the RNA polymerase. It binds tightly to these -35 and -10 sequences, and transcription is going to start a little bit down from this. So once the sigma factor is bound, RNA polymerase unzips the DNA and it starts making an mRNA copy. The sigma factor will fall off and RNA polymerase will continue to chug along using the template strand to build the mRNA here. Okay, so sigma factors are interesting. Promoter regions may have different -35 and different 10 sequences. So here you can see this -35 is TTGACG. You don't have to know this, but I'm just showing you. And the -10 is TATAAT. There is a certain sigma factor that will recognize these sequences. There are different sequences for different sigma factors. So in this case, this sigma factor recognizes this and goes, oh, there's a gene here or a set of genes that we need to transcribe. But different genes get turned on at different times. So there are actually many different sigma factors that are found in bacteria. So there might be a sigma factor that responds to heat stress. Okay, the bacteria gets heated up and it needs to express certain genes to deal with that. Or the bacteria needs to start dividing. There's a sigma factor that finds genes for division. If it needs to bring iron into the cell and metabolize it, there's a sigma factor for that. If there are too many bacteria around and the population needs to go into what we call stationary phase, where they slow down division, there's a sigma factor for that. So different conditions stimulate different sigma factors to be made. Now, sigma factors are protein, so there are sigma factors that control the expression of sigma factors. It gets very inception very early on, but you can start to see that this is like a little computer program almost. And I'm going to tell you that computer programming, computer science, and gene regulation are very closely related. We're going to talk about feedback loops. We're going to talk about different expression patterns and things like this. So if you understand computers, you're going to have a good time understanding gene regulation. So these sigma factors are one of the mechanisms of gene regulation, which is controlling what genes get made and when. So not all genes are made all the time, right? That makes sense. I talk about it this way. Cells need to save energy, right? There's a limited amount of resources in the environment. Cells don't want to waste energy. Think of it this way. If you're a bacteria and you can normally do glycolysis, processing glucose, you're going to do that unless you have no glucose around, right? Then you might need to switch to another sugar. So E. coli has the ability to ferment lactose, okay? But if there's no lactose around, it's useless to make the genes to break down lactose. So we're going to turn those genes off until the conditions are right, okay? So saving energy. Different sigma factors then are going to be expressed under different conditions. There might be one for, say, lactose, when lactose is present. So you might find in front of genes different -35 and -10 sequences so that different sigma factors bind under different conditions. And this whole thing, understanding promoters and even manipulating them to make our own promoters, is a burgeoning science, right? That's really cool. You can engineer a bacteria to start making a protein when you tell it to. I think that's super cool. And I will show you an example of that in the lab too. Don't worry. Okay, so sigma factor is part of the RNA polymerase control of transcription. So let's practice transcribing here. I have given you a sequence of DNA. I have told you which strand is the coding strand and which strand is the template strand. It is now your task to transcribe the RNA and transcribe everything you can. Remember the rules of RNA, just like the rules of DNA. Okay, go and pause the video and give this a try. Transcribe the RNA from this DNA. Okay, so the template strand is the one that we're going to use to build the mRNA. So we're going to start over here and we're going to start transcribing off the template. So we're going to use complementary base pairings. T pairs with A, G pairs with C, then AA. And here's our first difference. We have U instead of T, and continuing on in a 5' to 3' direction until we reach the end. Okay, that is how we transcribe RNA. Where this happens and when this happens, we're not going to worry about. So like if I give you an example like this, you're just going to transcribe where I tell you to transcribe, which is the whole thing in this case. Okay, so the steps of transcription. Well, there's an initiation phase, right? Sigma factor joins up with the RNA polymerase and they scan along on the DNA to find that promoter. Once the promoter is found, RNA polymerase binds tightly to the sequence. Sigma factor falls off and we start transcribing. We open up the DNA and start adding the RNA nucleotides in there. That's what RNTPs are. Elongation is going to continue until we reach a termination sequence. I did not discuss these because they vary drastically. In prokaryotes, there is some consensus that a lot of times what happens is there's a sequence that makes the RNA fold back on itself. And this causes RNA polymerase to kind of stutter or pause, and then it will fall off. What that sequence is and whether it's the same between different species, that's a complicated question. It tends to vary by species. This is how termination occurs in bacteria. How it occurs in eukaryotes, complicated as well. So Those are our steps to transcription. And at the end of transcription, we will have an RNA. We're going to focus on mRNA in this case. Okay, so the sigma factor binds to a region upstream of the gene known as what? Go ahead and pause the video. Think about that. Okay, the sigma factor is going to bind to the promoter region, which is upstream of the gene. The sigma factor is an example of a transcription factor, but it's going to bind to the promoter. All right, so that's transcription in prokaryotes. RNA polymerase is the enzyme that transcribes DNA into RNA. RNA is single-stranded. It contains U instead of T. That's sugar ribose in there as well. Of the two DNA strands, we have one that is the template. That is the strand that is used to make the new RNA. The coding strand is the other DNA strand. It doesn't do anything. It just sits there. But when you look at it, it will read the same as the mRNA that's getting made. That's why we call it the coding strand. Many bacteria have multiple genes in a row in the genome, and we call this an operon. That operon all gets transcribed in one single linear RNA, but they will become multiple different proteins. That's kind of unique to prokaryotes. The sigma factor is one of the ways that gene expression is controlled in prokaryotes. The sigma factor binds to a promoter sequence upstream of the transcription start site. There are different sigma factors that are made at different times that recognize different promoters. This is a process we call gene regulation. The sigma factor is an example of a prokaryotic transcription factor. We will talk about more detailed transcription factors in eukaryotes in the next section. All right, that's it for 15.2.